So good morning, everyone, and welcome on the Friday coffee of the ELIXIR Czech Republic, where we present the services and tools coming from members of the ELIXIR Czech Republic Consortium. Many people in Czech Republic have the picking a mushroom as a hobby, and today we will show you when the hobby becomes an obsession, because Tomáš Větrovský from Institute of Microbiology is going to present the GlobalFungi database, which is, I think, the largest collection of the sequences of the genomes of the fungi all over the world. So Tomáš, please, the floor is yours. And if you will have a question, please use the chat.
Yeah, thank you for introduction. Welcome everybody. And I will tell you a little bit more about our database which was developed with many people, many collaborators. So it's quite a, I think, big thing. So on the screen you can see the first page of our web page. So it's just a summary of the content, and I will start to introduce what's inside. So okay. So what is GlobalFungi? GlobalFungi is a web-based interface, and we've tried to collect as many as possible some fungal markers from different studies, so which originated from next generation sequencing, so we can access to people some information, some content from studies which were already published, but after the study was done, those sequences are just laying in some repository. So we collect this data, and we try to dig out some information out of it. So we focus on fungi because our lab is doing a lot of with fungi and from various ecological ecosystems and habitats and so on. So, and we try to create something which is using this FAIR initiative. So data should be findable, accessible, interoperable, and reusable. So this reusability, I think, is quite important. So I would like to mention that our project is hosted on this ELIXIR machine. So we are very thankful to ELIXIR that we got the resources for it. So and the last thing is that this project is very living. So the database is growing all the time. So we also create some online submission systems. So everybody who publish some new data describing some fungal community can submit the data and the study to fungal database, and in next release probably his data will be included. So we publish it in this scientific data journal, and you can see that there were really huge amount of people on the paper. Most of them were collecting data, helping with some curation, because we went through all the studies and manually curate them. So what was at the beginning? We started with some study 2 years ago, which were describing soil samples from different studies. So it was a type of meta study, and after we published this paper, we were thinking what we should do with the data we collected, because it was quite a big effort to collect the data for the study. So we just continue with the collection and have an idea to create such a database. And as I mentioned, that our database is focusing on fungi. We are using right now only just ITS, which is some part of DNA, which is non-coding, and it's a part of ribosomal coding. It's next to the ribosomal coding genes, and it's located in the chromosomes as a repeat. So just for information for who is not so familiar with this type of marker. And okay, here is some scheme which is describing how the website is constructed, what is behind, what are the annotation sources. So on the right side you can see the gray part with all those studies. It's we started with investigating about hundreds or more than 1,000 right now studies from various journals and so on. So, and then we manually create them and choose only the studies which are using non-manipulated experiments, and we have quite strict rules which should be followed to get into our database. And then we use the data from public repositories, or we ask the authors for the data originated from the studies and combine them with this metadata from the studies, and annotate the markers based on Unite, which is another project from Estonia, focusing on taxonomical annotation and of fungi, and we used this database as a reference for species and genus and so-called species hypothesis classification. So with the metadata, we combine the GPS coordinates with bioclimatic variables, and all of those informations were concatenated in MySQL database, which is behind our website. And with the nucleotide sequences, we generated the representative variants. Now it's about 145 millions of ITS sequence variants in our database, and you can use the BLAST for searching between them. So you can also search for your favorite taxa just with the molecular sequence. So now I will show you some example how the database can be used. So this example is based on this Boletus edulis, which is nice edible mushroom. And in our database, you can choose for taxon search and search for example, species, this Boletus. And what you will get? You will get some info about taxonomy and how many samples are covering, or in how many samples are covering this species. And you can also see on the right corner that you can download all the species variants of ITS sequences which were annotated as Boletus edulis. And there are also many filters, and you can for example filter out only just sequences from forest or grassland, or some root samples. So then the next results, or so visual outputs will be somehow correlated based on this filtering. So this is an example of the result. So you can see that there are breakdowns of different sample types and biomes, and on the bottom part is the just everything which is in our database. So it's a breakdown of all samples, and up on the up picture or pie chart, you can see where this species was mainly found. So you can see this the soil samples were containing the most of the sequences of this species, and in another in the forest biome was the forest biome was the source with the most of the sequences. So it's quite obvious because it's forest mushroom. On this screen, we are using the information from the GlobalFungi database with the environmental variables. So we can see what is the window of for example, temperature and precipitation values or records where the samples were collected from. Yeah. So you can see that this mushroom is like some average temperatures, and the precipitation it's mostly on the wetter part of the world. Okay. Yes, and here is the same graph with the pH values, which are also from all the collected samples, and on the bottom part is again the information from the whole database, from all the samples. So you can see where it is to where it is shifted, for example, in that case. And on this screen is probably the most attractive visualization of our results. So you can see the locations of the samples and the abundances of the sequences from this species. So you can see it's mainly located around Europe, and some founds are also from North America. And on the left part, you can see that if you click on individual dot or this mark of representing the sample, you can see more details about every sample. So you can also download the sequences from the sample and investigate the study from which the sample was originated. So, okay. As I mentioned, it's not only the searching from exact species is available. You can also input your own sequence from your favorite, for example, fungi, or some unknown sequence, and you can identify it through the database. So if you put some sequence, in this case, I just randomly choose some ITS from some study, and I found out that it was found in our database, but and some unidentified fungi. But this unidentified fungi has some SH number, and as I mentioned, that there is this UNITE project which is trying to classify all known fungi, and this SH number is corresponding to some molecular taxa, which should be something like a species, but it's also provided for fungi which were never been collected as a fruiting body. And so every known fungi has this species number if it was in this UNI database or sequence. So UNI database is mainly based on Sanger sequences. So you can see the comparison if, for example, with Russula ochroleuca, that how is the distribution in the UNI database based on Sanger sequences and based on next generation sequences in our database. So it's fitting quite well. So you can have some check of how the database is working, and it seems that that's fitting quite fine. Okay. Yeah. Another tool in our database is this GeoSearch, and the GeoSearch allows the people just select the area of interest and get a list of all the species or SAGs located in that part. So you will get the list of selected samples and list of species hypotheses, and you can click through or went through different species hypotheses and see the location around the world. It depends what you are searching for, but it seems to be quite useful. Okay, and last tool is just the list of the studies and visualization, or you will get the results based on the study. So you will see the samples covered by the studies, and you can investigate for example, the species hypothesis within those studies and so on. Okay, yeah. We also create some video tutorials, so everybody who will go to the website, he can click on tutorials. It's on the main page also, and you can went through and see how to do individual task, what he's interested in. And this is the last slide. So here are some future prospects. We now, with our database, we have release number 2, which is the current, and it's containing almost 27,000 samples. So it's the current state, and we are preparing the release 3, which is now in progress, and probably we are getting close to the almost 40,000 samples which will be included in our database. So we will see how it will go and when we will be ready with that, but it's almost done. Okay, so I would like to thank you, and that's all from my side. And thank you also for to all of the collaborators and the people who helped us with that project, because I think it's quite nice work.
Okay, Tomáš, thank you very much for a very nice talk. There are 2 questions. So first is Maria Novotny, who asked, how often do you lack some information about sample? I was surprised that there was no category unknown in a source of data for boletus. Can you briefly answer it?
Sorry.
How often do you lack some information about sample?
How often
you don't have information about sample?
Yeah, it's I exactly don't know what does it mean, but we have some rules for the studies and for the samples which should be fulfilled to add the sample into our database. So all the samples with missing, for example, GPS coordinates will not be added there. I don't know if it is the answer for the question, but we should know at least several compulsory informations from each sample which is included in the database.
Because you are the deposition database, so you are requiring the information which must be fulfilled, right?
Yeah. And also, if for example, if you would like to see all the samples where this boletus was not found, you can also switch it on the map. So you can see the samples where it was not detected, but it doesn't mean that it was the fungi was not living on the place. It's just the positive observations, but we cannot provide a negative observation of the fungi.
Okay. Another question: how different are fungi generally? It means probably the shortest and longest DNA. And how long is the average DNA? And what is the proportion of coding DNA versus the length of the DNA? Is it known?
It's a question about genomes or this marker gene?
It's about the genomes.
Okay, yeah. Because I have some knowledge about the genome sizes of fungi, but it's varying very well, very much. So it's hard to like say in short, but there are some small genomes and really huge ones. So it's hard to say, but also our marker is very variable in the length of the region. So sometimes we are this is not really connected with the genomes, but for example, in our database we are missing a lot of fungi which have extremely large region coding this marker. So yeah, the differences are quite big, but I think I cannot say so much details about the individual cases.
Okay. Jan Jansa asking, do you encounter problem with comparability of some of the environmental parameters, or you only include those that are strictly comparable, e.g. measured by exactly the same method, like pH measured in water? Yeah.
This really good question. Yeah, we were fighting a lot with those things, and finally we were trying to put there only the values. We have two different types of variables. So some variables are some general variables which should be in the same metrics and always comparable, which are used for the graphs and so on. So for example, pH, we are using only the pH where the method was defined, so we can compare or recalculate somehow the pH on the same like measurement or unit system. And in some types of variables we just showing the original metadata from the studies, but we cannot create the graphs out of them. So yes, for some general purposes we are using the variables which are comparable.
Okay. Interesting question from Lucia Naimanova. Does the database contain strictly environmental samples? I looked just briefly and there are many microscopic fungi, some of them with potential to cause diseases. So do you also include human samples, for example?
Yeah, I have some break with the connection, but probably I got it. the question was if there are only just environmental samples, or we also included some, for example, samples from some human bodies or those things. Yeah, we didn't. It's not included there right now, but we are planning to put also the samples connected with the human bodies and sequences, maybe from also animals and some disease-linked samples. So it will be done, but right now it's only just environmental.
Okay, thank you very much. So I see no other question. So Tomáš, thank you again, and for the others, please meet next Friday, 9:30 at the Elixir Friday Coffee, and have a good day. Bye.
Yep. See you. Thanks.